Well, it's a bit of a strange story for biotech people, but I worked for Dow Chemical for 37 years, and a lot of that time was doing research for things like polyethylene, basically plastics. So I led that effort, and one of the things we were all interested in is nanomaterials, which we could maybe use in the products that I did help with, like Ziploc bags and Crocs shoes and salad bags. And we sent our request off to Central Research along with a lot of money, and 7 years later we got nothing. So after I retired, my partner and I, also who work for Dow, decided we were going to make nanomaterials work. And by golly, we did. We figured out how to take the key problem is when they make them, they're in bundles, and they're micron bundles, they're not nanomaterials. So we figured out how to do that, and we put them in batteries, we put them in tires, we put them in 3D printing, but all that's interesting, and one of the things we had hammered into us in Dow, worry about your tox properties. So we went off and worked with Charles River actually to find out, well, what is the toxicology of these materials? And it turns out we dramatically change it. They have no effects at the mice level, at the tissue level, at the cellular level, even at the DNA level, which was great. And then we noticed how interesting our tubes penetrated cells without causing any damage. Well, that led to the next thing is we can put anything we want on the surface of those tubes, and it would carry that cargo into the cell. And when it did that, then when it was in there, it would unload. Then the final piece, if you want, was over time the body would apparently, I'll call it, digest it. So what we ended up with was an extremely simple tube concept, if you want, because we didn't have to adjust the tubes at all. It's always the same. We adjust a little bit on the surface of the tubes by putting different surfactants, if you want, depending on the cargo. And voila, we had a transport system. And we can make this stuff by the ton, in fact do. And what we can do is take a small stream off, we call it molecular rebar, in the industrial side, and MGMR, medical grade molecular rebar, we're not that original, for the medicine. And so what we're in the process of doing, now that we've worked out most of the technology, is now looking for partners to develop it. Oh, CAR-T is an obvious one. Pretty much anything ex vivo, because ex vivo we can take and separate out the tubes with our material before it goes back in the body. Makes it even easier with regulatory agencies. But even with that, we now know we have a route. If we put it in the body, it'll eventually leave. And we have two people we're working with, two groups really, that are in vivo. Even though we said we'd like to do it ex vivo, they are the customer, and it's working out pretty well for us so far. Well, you totally bypass viral vectors. And what we really do, for our whole manufacturing process, it takes about 2 days. It's very complicated, extremely complicated. We put in a beaker with our stuff in the cargo, stir for 24 hours, and bottle it. Now, if we're doing the development process, usually that takes about a couple weeks, because we want to optimize that surfactant a little bit. So if you compare that to a viral vector, the time difference is totally different. And so you can get your material back to your patient much quicker. No, I think we're looking forward to facilitate and finding some more partners. And the more partners we get, of course, we have the big obstacle, which is tradition. Tradition is viral vectors. Well, we're something that's not viral vectors, so we need some people who want to try it out and see, okay, it really does work. And so far, everything we've tried seems to work. It's great.